Alright, Assalamualaikum and very good morning to all my lovely students. Hopefully today you are all in healthy and fine condition. Alright, so let's continue our discussion on cuti-cuti belajar handout on the chapter 6. Alright, so the title of the chapter 6 is cell division. Okay, so now we start with the question on the section A, question 1A and B. Okay, let's take a look on the question. Diagram 1.1 shows a micrograph of cross-section tips of onion root. Okay, so this is referred to the micrograph of cross-section tips. Yeah, cross-section tips of onion root. Yeah, it is referred to the tips of onion root. Okay, so you have to identify the diagram given here. Yeah. Next, we go to the question 1A. State the type of cell division of cross-section tips of onion root. So as we had learned in the form 4, okay, we had learned that cell division can be divided into two types. Okay, what are they? Mitosis and meiosis. Okay, so you have to identify, is it mitosis or meiosis occur here? Right, so the clue given here refer to the tips of the onion root. Yeah, tips of the onion root refer to the meristematic tissue. Yeah, meristematic cells or tissue. What do you mean by the meristematic tissue? It is a refer to the tissue. Yeah, tissue that actively or refer to the cells. Cells that actively dividing. Yeah, that is actively dividing by mitosis. Okay, so of course the answer is mitosis. Alright, okay, why mitosis? Because the process occurs in the tips of onion root, in the tips of root. So tips of root refer to the meristematic part of the plant. Okay, the other meristematic part of the plant is referred to the tips of the shoot. Alright, so at these meristematic parts, mitosis occur. Mitosis occur. That is referred to the cells actively dividing. Actively dividing by mitosis. Okay, next we go to the question B. Based on diagram 1.1, explain stage S. Okay, so let's take a look on the diagram here. So this is referred to the stage S. Yes, stage S here, as we can see, the chromosome align or line up at the metaphase plate. So this is referred to the metaphase. The stage S here referred to the metaphase. Okay, so metaphase. Okay, you have to explain what happened in the metaphase. Chromosome line up at the metaphase plate. Okay, chromosome line up at the metaphase plate. Okay, so you will get 2 mark. Alright, so if the question asking about this stage, so what is this stage? This is refer to the anaphase. Yeah, refer to the anaphase. Or here this is refer to the anaphase. This is telophase already. Yeah, it starts to divide. Alright, okay, so please take a look, please observe the diagram given, very carefully, to identify the stages in the diagram given. Okay, now we go to the question 1C. The diagram 1.2 shows a bryophyllum species with the plantlets growing at the leaf margin. Alright, so this is referred to the diagram 1.2. So as we can see from the diagram here, this is referred to the plantlets. Plantlets that refer to the anak pokok, anak pokok. Alright, so explain how the plantlets are produced. Okay, so how the plantlets are produced. Okay, so you have to explain what are the process occur that will cause the production of the plantlet. Okay, first point, yeah, this is referred to the suggested answer. Plantlet undergoes mitosis. Yeah, so plantlet undergo mitosis. So mitosis will produce new cell. And the daughter cell or the plantlet identical to the parent cell. So the plantlet produced here are identical to the parent cells. Alright, so this is referred to the suggested answer on the question 1C. So you have to explain how the plantlet are produced. Okay, what are the clue given here? The clue given here refer to the plantlet, right? Okay, when it is referred to the plantlet that are related to the mitosis. Yeah, so how does the plantlet produce? It is starting from the mitosis process of the parent cell. All right. Okay, so right. So that is the answer for the question 1C. Now we go to the section A, question 1D, 1 and 2. Okay, diagram 1.3. A shows the cancerous cell which are formed after the normal cell are exposed to the factor W, and diagram 1.3b by the radiotherapy method. Okay, so this is referred to the diagram 1.3a, refer to the cancerous cells. Yeah, cancerous cell here, which are formed after the normal cell are exposed to the factor W. Yeah, okay, so when the normal cell are exposed to the factor W, it will form the cancerous cells here. And then the diagram 1.3b here refer to the radiotherapy method. So this is referred to the radiotherapy method, okay, whereby it is a method by using a radioactive ray, yeah, radioactive ray to destroy the cancerous cell. Okay, now we go to the question 1. Give one example of factor W. Okay, just one example of factor W. You can either give radioactive ray, or you just can write UV ray, ultraviolet ray, yeah, or any ray, yeah, alpha ray, gamma ray. Okay, and then you can also write the chemical substance, yeah, chemical substance, or you can write the mutation. Right, so this is referred to the example of the answer for the question 1, D1. Okay, now we go to the D2. Why radiotherapy is used to control or stop the growth of cancer cells? Okay, why radiotherapy is used to control or stop the growth of cell? So you have to explain, okay, explain about the radiotherapy method. Okay, so this method uses a high power ionizing radiation. Yeah, okay, high power ionizing radiation, or you just can write that use the radiation to kill the cancer cells. Yeah, to kill the cancer cells. Okay, high dose administration is administered to the target or to the tumor, yeah, to heal, control, or reduce symptoms of the disease. Okay, so that is referred to the explanation on the radiotherapy method. Okay, now we go to the section C. Yeah, section C, 20 marks. Right, so this is compulsory section that you need to answer in the essay. Okay, question 1A, 1 and 2. Okay, 1A. Stem cell therapy, also known as regenerative medicine, promotes the repair response of disease, dysfunctional or injured tissues using stem cells. Alright, that is the introduction about the stem cell therapy. Yeah, the use of the stem cell therapy. Yeah, which is referred to the promotes the repair response or promotes the dysfunctional injured tissue using the stem cells. Okay, so as you can look from the diagram here, this is referred to the stem cell. Yeah, Stem cells that are able to differentiate to any, any cell, to any cell, all right. Okay, diagram 1.1 shows the potential application of human stem cell, potential application of human stem cell. Okay, so in one, based on diagram 1.1, give the meaning of stem cell. So what is the meaning of the stem cell? So stem cell refer to the cell. Okay, this is refer to the suggested answer here. Refer to the cell that can be divided. It can be divided into new cells of the same type. And the cell that can be divided into the new cells, okay, by mitosis. And then this cell will differentiate, differentiate into other types of cells to carry out specific function. Okay, at first the cell divided into new cells. Okay, this stem cell able to divide into the new cells, and then after that it will differentiate into other types of the cell, to carry out specific function. Okay, that is referred to the stem cell. So that's why stem cell therapy is very important now, because the stem cell is able to divide, divide into the new cell, and it also able to differentiate into any types of cells, to any type of cells to carry out the specific function. Okay, that is referred to the 1A1. Okay, now we go to the question 1A2. Okay, leukemia is a blood cancer. Leukemia is a blood cancer where too many white blood cells are produced, and these white blood cells do not mature normally. So these accumulate in the bone marrow as well as in the blood, and also suppress the formation of the normal blood cells. Okay, so the production of excessive number of these cells in the blood and the loss ability to fight infection is what makes the leukemia rapidly fatal if not successfully treated. Okay, so this is referred to the leukemia. Leukemia disease, which is referred to the blood cancer. Referred to the blood cancer, whereby there are too many white blood cells produced, but these white blood cells cannot be used, or they are not mature normally. And it will cause the white blood cell cannot function. All right. Okay. So by using your biological knowledge, justify whether stem cell therapy is suitable to treat cancer like leukemia. Okay. So by our knowledge on the biology, so justify. Okay. So justify means that you have to analyze. You analyze whether it is suitable or not. Okay, if you think that stem cell is suitable, you have to give the point to support your choice. Okay, so you have to choose whether you agree or not. Agree or not, a stem cell therapy is suitable to treat cancer like leukemia. Okay, so since we have discussed about the stem cell therapy, which is able to divide into new cell and differentiate into other types of cell, to carry out a specific function, so the answer is referred to the agree or yes. Yes, stem cell therapy is suitable to treat cancer like leukemia. So when you answer or you write yes or agree, that is the first point. Okay, that is your choice. Okay, and next we go to the point how to support your choice. Okay, stem cells are collected from the blood or bone marrow of a healthy donor. Healthy donor. Okay, you have to collect the stem cell from the bone marrow of the healthy donor, and also you can get from the umbilical cord blood. Umbilical cord of the baby, of the embryo. Okay. Next, the stem cell can differentiate into healthy or specific blood cells, and then it will replace dead cells damaged by chemotherapy or disease. So the stem cell also contain immune cells to fight leukemic cells. Okay, that is referred to the suggested answer for the question 1A2. It is about the leukemia. Alright, so by using the stem cell therapy. Okay, next we go to the question 1B1. Yeah, 1B1. Diagram 1.2a shows a method of growing a type of plant in farm A. And diagram 1.2b shows a different method of growing the same plant in farm B. Alright, so this is referred to the diagram 1.2 that show a type of plant in farm A. So this is referred to the type of plant in farm B, yeah, that use a different method of growing, right? So this is referred to the farm A, right? This is referred to the farm B. Okay, so you have to analyze the diagram, yeah, carefully. All right, so you have to identify the method, yeah, method use of growing the plant, yeah, in farm A and in farm B. So we go to the question 1. In your opinion, which farm will generate more profit in next 10 years? Give reason for your answer. Okay, so you have to underline, yeah, what the question wants, yeah, from here. Okay, so which farm will generate more profit? So you have to underline which one will generate more profit in next 10 years. So when it's asking about it's about the next 10 years, it is referred to the long duration of time, a long duration of time. All right, so in next 10 years. All right, so before that you have to identify the method, yeah, method of growing of this plant. So this is referred to the traditional method whereby the plant is planted in the soil. Okay, and the plant will undergo sexual reproduction and will produce a new plant. All right, and then but here, so this is referred to the tissue culture, yeah, tissue culture method. Yeah, so tissue culture method is a method to produce a lot of the plants in the short duration of time, yeah, in a short of duration of time. So you have to decide which form, yeah, which form will able to generate more profit in the next 10 years. Okay, is it method A or method B? All right, so in the question, from the question, so what is the suggested answer here? It is referred to the farm A. Yeah, farm A. So this is referred to the plant that use the traditional method, yeah, in the farm A. Okay, so the plantlet are produced by sexual reproduction, which involve fertilization of the gametes. Yeah, that involve the fertilization of the gametes. All right, so when the plantlet is grow into the adult plant, it will produce a flower. That flower will contains the gamete, and then the fertilization occur between the gametes. So that is referred to the sexual reproduction, and then it will produce the new plants. And there are varieties of plantlet are produced, so it will cause the production of the variety of the plantlet. Right, so therefore the plantlet have resistance, yeah, have different resistance toward the different disease. Okay, so the plantlet have resistance towards a different disease because the plantlets, in every plantlet is not the same, so it will resistant to the different disease, so it will able to adapt to the changes in the environment, and this will ensure the plantlets can live longer. Okay, that is referred to the suggested answer for the question B1. Yeah, so if you choose diagram 1.2b, which is referred to the tissue culture method, you also can choose it, but you have to give a reason. Yeah, reason and relate. Yeah, relate how it able to generate more profit in the next 10 years. All right, okay, so so in this question, in this question you are suggested to choose farm A. All right, okay, Next, we go to the section C. Question 1B1 and 2. Yeah, the farmer who used method in diagram 1.2b found that all plants died after benign attack by a certain disease. Okay, then suggest ways to be taken by the farmer before he starts the method in diagram 1.2b in the future. Alright, so we have to suggest to the farmer, yeah, that use the tissue culture method. Yeah, so what our suggestion? Right, so this is repeated suggested answer. First, choose a plant that is resistant to various type of disease before we culture it. Yeah, we have to choose a plant that is resistant to various type of disease. Or we also have to choose a plant which have been genetically modified. All right. So usually the modified organism, the modified plant are already resistant to the certain disease. All right. So we have to choose a plant that which have genetically modified that will produce the GMO, genetically modified organism. Okay. Next, choose a plant that has high resistance to insecticides or pesticide. All right. So that is a waste, yeah, that can be taken by the farmer, yeah, before he start the tissue culture method. All right. Okay, next we go to the question 3. Diagram 1.2 shows a group of orchid plants which are genetically identical. Okay, genetically identical. It also produced through methods 1 to B. Okay, 1 to B just now refer to the tissue culture method. Okay, based on the diagram above, describe the techniques used to produce this plant. Okay, so you have to explain about the tissue culture technique. Okay, how do we know that is a method which is tissue culture method? Because they are genetically identical. Yeah, genetically identical. Yeah, so they are same to each other. Right? Okay. So how to describe the techniques or to explain the techniques? Okay, let's go to the suggested answer here. So you have to state what is the technique or what is the method. The method refers to the tissue culture. Okay, the tissue culture method or technique. Okay, so how to carry out the tissue culture technique, you have to explain one by one. Okay, first, the leaf or shoot or stem or root tissues from the parent cell are cut into smaller pieces that called as explant. Yeah, cut into smaller pieces or explants. So the pieces of explant are cultured in a sterile nutrient medium. Okay, we have to put the explant in the culture medium, which is sterile nutrient medium. And then the temperature and pH medium also maintained at optimum rate. Yeah, the temperature and pH medium are also maintained at optimum. The cells divide by mitosis. All right. So what happened to the explant? The cell of the explant will divide by mitosis to produce callus. Okay, produce callus, and then the callus is then grow into embryo. The embryo then will develop into plantlet, right? From the embryo, will develop into the plantlet, and when it has grown into a suitable size, yeah, the plantlet just now is transferred into the nursery, yeah, to the nursery or to the soil. All right. Okay, so that is referred to the tissue culture technique. All right. Alright, okay, we go to the last question on the section C, which is 1C. Diagram 1.3 shows a karyotype of a person with chromosome abnormality. Okay, so this person have chromosome abnormality. Yeah, please identify the diagram. So you have to identify how this person is abnormal. Yeah, okay, so as we can look here, on the 21st chromosome, there are 3 chromosomes. That is abnormal. Yeah, 3 chromosome or extra chromosome on the 21st pair. Yeah, if normal, it's just only have 2 chromosome. Yeah, on every pair. Okay, so we go to the question. You are asked to explain the health issues on the chromosome abnormality shown in the diagram 1.3 to the pregnant mother. Explain this abnormality. Okay, so what is the type of abnormality? If we go to the diagram again. So when there is an extra chromosome on the 21st pair, okay, this abnormality will cause a Down syndrome. Yeah, Down syndrome. Okay, so first point is Down syndrome. It is a type of genetic disease. Okay, it is a type of genetic disease. Yeah, so means it can be inherited. Then, okay, an individual with Down syndrome has an extra chromosome at the 21st site, or we call as trisomy 21. Right, so the individual has an extra chromosome at the 21st pair of the chromosome, or you just can write trisomy 21. Abnormality during division of the meiosis. Yeah, okay, so it is occur because the abnormality that occurred during meiosis. Okay, what happened? Spindle fibers fail to function during anaphase 1 or 2. Okay, as a result, chromosome fail to separate. Yeah, as a result, chromosome fail to separate or non-disjunction occur. Alright, so we already discussed the non-disjunction, yeah, in the last 2 chapter, yeah, in the form 5, right? Okay, the gametes, yeah, the gametes have an abnormal number of chromosomes. Yeah, the gametes have an abnormal number of chromosomes, either 21 or 24 chromosomes. Okay, fertilization between the normal gamete, which is 23 chromosome, and abnormal gamete that which have 24 chromosomes. Okay, so it will cause the production of abnormal zygote, yeah, with 47 chromosomes. Yeah, so when the fertilization between the normal gamete and abnormal gamete that have 24 chromosomes, so it will produce the abnormal zygote, which have 47 chromosomes. Okay, the normal zygote is only has 46 chromosomes. If abnormal here, it will cause the number of the chromosome of the zygote is 47. Yeah, so the characteristics of this disease are mental retardation, squint eyes or slanted eyes, yeah, sticky tongue, okay, broadened broadened neck. Yeah, all right, so that is referred to the symptoms, yeah, of the Down syndrome person. Okay, so we already discussed all the question in the chapter 6, cell division. All right, so please study more about the cell division, especially the stages in the meiosis, yeah, the stages in the mitosis. So you have to differentiate between these 2 types of the cell division. All right, okay, so I think that's all for now. So please take care everyone. Okay, stay safe everyone. Thank you.